Welcome everyone to another Build a Cell seminar, and thank you, Yujia, for agreeing to give a talk today. I will hand it on over to you to introduce yourself and then take it away.
Thank you, Carlos. Thanks for the invitation. And thanks everyone for joining this seminar. It's my great honor to present in this Build a Cell seminar. And today my topic is Dropletronics, which later I'll explain what does it mean by this awkward term, Dropletronics. But it's about microscale droplet assembly enables biocompatible, multifunctional, and modular iontronics. So to begin with, you should be able to see my second slide. Hope that's the true. So I'm at École in Switzerland, Lausanne. It's a small town but beautiful with the Geneva Lake. Our lab is very young. We called ourselves the Laboratory for Bioelectronics, BION. We just started in January and on the third floor of the central BM building, but we are growing fast. We have 3 students at the moment, 1 postdoc, and more students to join us. And also we welcome other students who are interested in our labs and postdocs later on. If you have any questions, just email me directly. I'm happy to answer them. For myself, my name is Yujia Zhang. Previously, I was at Oxford in the UK. I did my postdoc there and later became an early career fellow as well. Before that, I did my PhD in China, in the Chinese Academy of Science in Shanghai, and I did a collaboration project with NYU and Stony Brook at the US. So back to my group, we are highly interdisciplinary. Later on, you will see the detailed research we are doing. So it covers manufacturing, bioengineering, materials, energy, electronic engineering, and a lot of biological works. And the vision is we believe is a big vision. Our mission is to make bioelectronic systems for biointerfaces and hybrid intelligence systems, such as neuromorphic computing, some iotronic computing as well. So the outline today, briefly, I'll introduce the meaning of our research and what is Dropletronics. And then I will give some examples about droplet batteries, hybrid printing, and especially one latest, the most recent publication we did last year, by the end of the last year, published in Science, is about this dropletronic diodes, circuits, and bell interfaces. And later on, I will summarize my research with some future-looking perspectives. So, what is Iontronics? So last year, basically, based on this international chemistry organization, they summarized the top 10 emerging technologies in 2024. And at the bottom left row, you will see this nanofluidic iontronics. And basically, I believe they used a wrong schematic to represent this field. So actually, we should put some aqueous solution here because for nanofluidic iontronics, it is about the movement or transport of ions and small molecules, such as neurotransmitters. And fundamentally, it is mainly in aqueous solutions, which is very different compared to electrons in solid state materials. So for electronics, it's mainly in aqueous solutions and about the transport of ions and some charged molecules or even organic compounds. So moving on to the benefit of iontronics. So fundamentally, iontronics shows its major benefit in the bell interface or at the bell interfaces. So because if you think about it, iontronics and our natural cells and tissues use the same language of ions. And fundamentally about bioelectricity is actually about the transport of ionic fluxes and some charged compounds. So if we use electronics to interface with cells or tissues, we usually face this high interface impedance, so which is a problem. So basically, we will have 2 cases. Either we create this electrical double layer between the metal electrode and biological tissues, or if we input very high input current or voltage, we create this electrochemical reactions and created this ion flux in the biological environment. So basically, we will create this high interfacial impedance, and therefore we limit really the biocompatibility of the devices. Let me give you a detailed example. For example, we all know for neurons to fire, the membrane potential is around 10 to 100 millivolts. However, if you check the literature or in some practical cases, the actual input voltage for spinal cord stimulation or deep brain stimulation, usually the input voltage is about 5 to 10 volts, and clearly is about 100 times or even higher compared to the native membrane potential. So if we could use ions to directly interface and stimulate cells or tissues, that would be a clear benefit in comparison with electronics. However, I need to highlight that the current challenge for iontronic field is the integration level of the device is quite low, especially compared to the highly integrated electronic circuits. The reason for that is because at the moment, most iontronic research is only focus on the materials, for example, hydrogels, some conductive ion polymers, etc. But people rarely focus on the integration of those devices and to fabricate those iontronic miniaturized systems. But on the other hand, we do witness like an emerging field, so-called nanofluidic iontronics, which people use basically biological nanopores or artificial nanofluidic components like carbon nanotubes, some 2D nanoslits to create this unique, and people found unique phenomenon when we use nanoscale components to control the ion transport, etc. But the real problem is obviously there's a missing gap, right? Can we shrink down those large scale hydrogel devices and also incorporate some nanoscale fluidic components? And that is where my research and our group focus on. So previously at Oxford, in Professor Hagen Bailey's group, we developed this droplet technology, and it's actually very simple. So the rationale is that we deposit aqueous or hydrogel droplets in surfactant containing or lipid containing oil, and those lipid or surfactant molecules will automatically cover those aqueous droplets and form this monolayer. And when we have multiple droplets in contact with each other, they will form this droplet interface bilayers, basically two layers of lipids or surfactants, and which mimics the natural droplet membranes. And we can also incorporate nanofluidic components in the aqueous droplets, or depending on the material, we can also incorporate them in the oil phase. And those nanofluidic components can automatically insert into those DIBs, droplet interface bilayers, and form this droplet-droplet connection, which is very useful in controlling the ion transport, molecule signaling, etc. And also we have this droplet printing technique, and we can deposit thousands of those aqueous droplets together to form this droplet network, which not only structurally but also functionally mimic the natural tissue, our brain, for example. And the goal is actually to achieve functionalities go beyond mimicking nature. Later on, I will show you some batteries, some computational work so we can do with this droplet networks. And this is the droplet printing technique I showed previously. This was developed in Professor Bailey's lab, and we're at the moment optimizing this system. So the mechanism is very simple. We have this glass nozzles in connect to a piezo actuator, and the actuation of those actuators will push out or squeeze out those aqueous droplets in the lipid-containing oil, and we can have multiple nozzles working together to form a droplet network. The resolution we can achieve can reach to tens of micrometers and can also be very large if we want to do some cell printing. For example, we can also incorporate fluorescence labeled cells into those droplets and form this beautiful network. The way we use those droplets to build iontronics is summarized in this slide. So we have two pathways. So after depositing aqueous droplets, either we keep them as aqueous solutions, so basically we do not gellate them, and therefore those membrane channels can insert it into the DIBs that is kept in this scenario, or we trigger the gelation of those droplets by, let's say, UV cross-linking or temperature cross-linking, and therefore the gelation of those polymers will rupture the internal droplet interface bilayers and form this continuous hydrogel droplet network later on for other, let's say, larger battery output or hydrogel network for other applications. So we can either use nanofluidic channels or we use the hydrogel network with embedded materials and different hydrogels to achieve different iontronic functions. And fundamentally, we can achieve this in 3D scale to build this droplet network and achieve hierarchical functions. So I'll give you a quick overview of some examples we have been doing and start from this droplet battery, which is, I think, quite elegant work. Basically, we mimic the energy generating mechanisms of electrical eel in nature, which is a quite powerful animal in the sea, in the ocean. And this electrical eel use purely ionic transport to generate output power. And the way to do that is it can use this asymmetrically distributed cation channels, for example, sodium channels and potassium channels to translocate sodium and potassium across the cell membranes and achieve this purely ionic-based output power. And the question for us is, can we mimic this structure and also use ions as an output energy source and to achieve some biomedical functions? So that is the central goal of this project. And the way we did it was just by depositing droplets, as I mentioned in my last few couple of slides. So the way to do that is very simple. So we deposit 5 droplets for one salt gradient battery. On the two ends, we have 2 droplets with high salt concentration, and at the middle, we have a droplet with low salt concentration. So basically, we have a salt gradient from the two ends to the middle, and in between, as shown here in the yellow and the green droplets, we have a cation selective polymer and an ion selective polymer. So basically, only cations can move from left to the middle, and only anions can move from right to the middle. And therefore, by combining this together and trigger the gelation, which ruptured the IBs, we can form this continuous hydrogel network. And those, because as you can see, cations move from left to the middle, anions move from right towards the middle. We have a directional movement of all ions and can generate this ionic output power. And the beauty of this is we can store all those droplets when we have those DIBs, and we can keep this structure in, let's say, an incubator for a couple of days, which is very stable. And when we want to use this battery to generate output powers, we can trigger the gelation and immediately the output is on. So it's like a switch from off to on. It's like a triggering process. So here are some basic outputs of the droplet battery. This is the photo showing actually the structure. The color is I need to mention it's from colorful dyes. It's not the original color of those polymers. And we can deposit very small droplets. In this case, each droplet is about 100 or 200 micrometers. And the whole network can be encapsulated into, let's say, an organogel, which is flexible and stretchable and even compressible. So we can make this droplet battery portable, and later on I'll show you how we use this to simulate cells and tissues. And for a single droplet battery, the output voltage is about 100 to 150 millivolts. The output current is about a couple of microamperes, 2 microamperes in this case. And it could last for about 30 minutes to a couple of hours once we switch it on. When it's at the off stage, we can keep it for hours. And once it's switched on, it could last for 1 or 2 hours. And also, the benefit of using droplets is because droplet is modular, meaning that we can combine multiple droplet batteries together. Let's take a look at this case. For example, if we put 2 batteries in series or in cascade, then we can double the output voltage. If we put them in parallel, then we can double the output current. And later on, we can put tens or hundreds of them together, and we can actually use this to power up like an LED or some other electronic circuits like a pulse generator, etc. But the most important or actually unique application we can do is to use this droplet battery to stimulate or modulate cell activities. And in this case, we use neuronal organoids as an example, and we combine the neuronal organoids into some droplets, and we combine the droplet with our upper, as you can see here, the droplet battery. And when the output ionic current pass through the neuronal organoids, the ionic current will modulate the neuronal activities. And in this case, we use intracellular calcium dyes to show to investigate the neuronal activity. As you can see here on the right-hand side, the ionic current passes through the organoids from left to right, and we do see a corresponding calcium waves generated inside of those neuronal networks. And importantly, if we compare a very young neural organoids, let's say 3 days old, and slightly matured neural organoids after 2 weeks, they do show a significantly different response. Younger neurons are more separated, and for a network, when they form this network, they're connected, and therefore they show this network level response. And this is just the first example I want to show you today. And when we published this work in Nature and also highlighted by Nature Methods, we were already thinking about can we use the same mechanism in terms of generating those hydrogel droplets or dropletronics, and can we use other energy generating mechanisms? Can we interface with other biological constructs such as actual organs, which necessitate a higher output and power? Also, can we achieve other iontronic functions? And later on, I will show you some examples to solve each of those questions. So first, of course, we can use other energy generating mechanisms because those droplets fundamentally, I believe, is a versatile platform, right? It's hydrogels, and we can incorporate the hydrogel with non-fluidic channels, with other particles, etc. So in this case, we incorporate lithium particles into the hydrogel droplets, and we build this lithium-ion hydrogel droplet batteries by the same manufacturing process. So we deposit droplets in the surfactant containing oil, use UV light to cross-link it, and they form this continuous hydrogel network. And at the two ends, we have lithium cathode and anode, and at the middle, we have a drop hydrogel separator to separate those electrodes together separately. And we can achieve, by shrinking down the droplet volume, we can do a very miniaturized lithium battery. And by comparison to other soft lithium-ion batteries, we were the smallest lithium-ion hydrogel batteries and also has the highest energy density because we can really print a miniaturized droplets and with high particle density. And because of the structure and the small size of those droplet batteries, we can do a lot of different applications. So the first case we did is to connect the lithium droplet battery with synthetic cells. In this case, aqueous solutions in the droplet forms and connected by DIBs with alpha hemolysin membrane channels. And the output of on the left-hand side, you see the lithium droplet batteries, LIDB, can power the charged ions or charged molecules to translocate through those membrane channels. In this case, let's say ATP molecules through alpha hemolysin channels. And this is widely controlled with directional movement because of the power of the ionic current. Also, we can incorporate magnetic particles in the middle separator or in the middle droplet. And as you can see here in this video, we can use an external magnetic or magnets to move this droplet batteries. And in some applications, we can also translocate this from a charging electrode, recharging electrodes to target electrodes to deliver this energy as a sort of energy courier. But the most important application we did was to use this quite powerful output to stimulate or actually defibrillate an ex vivo mouse heart. So we collaborated with Professor Ming Lei in Oxford in pharmaceutical department, and we created this ex vivo mouse heart model. And when we deposit this lithium-ion droplet battery simply by using a pipette, and this output power can basically shock the mouse heart. And this is also correlated to the droplet size, because if we have a larger droplet, it will have more contact area and therefore a stronger stimulation. But also, even for a very small, tiny droplet battery, we can also already achieve a stimulation. And this could be powerful enough in terms of defibrillating a mouse heart. So in this case, we've first treated the heart with some drugs to toxicate it, and then we use the droplet battery to defibrillate the heart, and then we see the heart rapidly within 10 seconds, and it can go back to the normal heart rhythm. Beside lithium particles, we can also incorporate, let's say, metabolic reactions. This is a more, I guess, similar to the general topic of this seminar, synthetic cells. So we can incorporate, let's say, the oxidation of an ADH molecule in the droplet, and the oxidation will generate involves the charge transfer, and therefore it could be used to build this droplet-based fuel cells, and it could be used to power again ion transport and also charged molecule transport if we combine this droplet fuel cell with a droplet network. In this case, it can be used to translocate a charged powering Y into the target droplets, depending on the distribution of the cathode or the anode droplet. We could also incorporate cells into the droplets to achieve bioprinting. And in Oxford, I co-supervise these two students, and they demonstrate a lot of different examples. For example, we can use droplet to print tumor cells, to form brain organoids, and to form cardiomyocytes, tissues, etc., etc. So I would say the droplet platform is very versatile, and we can use it to do many different things. And Iontronics is the current focus of my lab here at the EPFL. And later on, we were thinking of how do we control the generated ionic current. So previously, we have droplet batteries, right? So we can generate output purely by ionic flux or ion transport. The question is, how do we control the generated ion signals? So in this case, we were thinking about how electronics can control the movement of electrons, and it's based on these p-type or n-type semiconductors, and basically by doping a silicon wafer with different ions, right? So maybe we can achieve the same thing with hydrogels, and in this case, we did mimic the p-type and n-type semiconductors by using just cation selective hydrogels and anion selective hydrogels. So if it is the polymer is negatively charged, basically have some, let's say, amino acid residues, which is negatively charged, then the polymer hydrogel could be seen as cation selective. So only cations can pass through them, and which is basically mimicking p-type semiconductors. And vice versa for anion selective hydrogels is mimicking the n-type semiconductors. And in this case, we use silk fibering as the polymer to build this cation or anion selective hydrogel. Because silk fibering has so many different benefit points, it is biocompatible and is natural polymer, but most of all, it has so many, it has a rich composition of those amino acids, and which can be used to achieve further chemical modifications. And in this case, we first link the silk fibering, which is at the bottom, with hyaluronic acid termine conjugate, which is very negatively charged, and therefore we can create this cation selective silk fibering hydrogel. And on the other hand, we can also link silk fibering with a positively charged polymer such as polylysine, and also can be further modified to make it even more positively charged. So therefore, this polymer will be anion selective. So if we see the right-hand side, the original surface charge of pure silk fibering is the black curve, which is a slightly negative charge. We can enhance it or reverse it to cation selective or anion selective. So and also eventually, because of silk fibering has this dityrosine or tyrosine on it, we can use UV crosslink to form dityrosine bonds, which will crosslink the silk hydrogel. And by using the same surfactant-supported droplet printing, we can deposit those silk fibering hydrogels in the lipid-containing oil. And in this case, it's very simple. So if you just have two droplets, they will form this droplet diode. If you have three droplets, we will form this transistor. I'll show some characterizations later. And by optimizing the printing technique, we can actually shrink down the droplet to about tens of micrometers in diameter for a single droplet, or in total about 200 to 300 micrometers for a transistor. So for the Dropletronic diode, it's very simple. It's just combining a cation selective and anion selective droplet together to form this bilayer, a droplet bilayer. And it is only conductive from one direction to the other, because the reason for that is there is only limited mobile ions at the interface. So if it's just from one direction, those mobile ions can pass through the interface. But if we apply a reverse input, as you can see here on the left-hand side, those mobile ions will be depleted, and therefore we will see a very limited conductive current. But on the other hand side, if it's conductive, then we see a much higher conduction. And this, because of the small size of those droplets, there are only very limited. This is only limited amount of those mobile ions, and therefore the response time is actually very fast if we compare to previous iontronic, larger scale iontronic devices, as you can see here. However, the main challenge we had was because of the small size of those aqueous or hydrogel droplets, the waters are very easy to evaporate if we just keep the device in a room condition. So let's say for this, after 24 hours, because of the water evaporation, we do see the significant increase of the resistance of those systems. However, we can restore partial of the performance by rehydrate the droplets by, let's say, pipetting a small aqueous droplet around it to rehydrate the droplet, or by placing the Dropletronic diode on the water-saturated substrate, let's say silicon dioxide substrate saturated with water. And also we verified the conduction by impedance analysis, and as I mentioned, the response time is very fast. Moving a step forward, beyond iontronic diode, what is interesting is the iontronic transistor, and the working rationale is actually the same. So we, at the middle gating droplet, if we, let's say, apply, in this case, an upward voltage, it will push all the mobile ions back to the interface, and therefore it's more conductive. If we apply a reverse voltage or current, it will draw away all the mobile ions, and the conduction become less effective between the left and right collector and emitter droplets. It's basically like you open or close a water faucet or a water tap. If we apply, let's say on the right hand side in the data figure, we can achieve a very high on-off ratio, about tens to hundreds, depending on the droplet size as well, which is comparable to the state-of-art iontronic devices. And our system is much smaller than their larger scale devices. It also could have faster response because of the small size, as I explained. It could have, like, as you can see here, the response time for a single drop transistor is about 0. So tens or hundreds of milliseconds, which is sufficient enough later on, as I'll show you, to monitor, to sense the beating signals from cardiac myocytes. But actually, if we think about what we can do or the applications of this Dropletronic transistor, we should question ourselves, how do we think of using iontronics? Of course, we can use it as what electronics can do, which is to integrate them into, let's say, a Dropletronic circuit by putting multiple modules together and then form this connection. But more importantly, for myself, is to think in the iontronic way, which is, can we try ions? Because ion has so many different species and differentialities. So can we try ions with different sizes or even different ions, right? And also, can we use biosignals as the gating signals to modulate the middle droplet? Because biology also uses ions as the signal carriers. So later on, I will show you 3 applications, what we can do with this Dropletronic components. But more importantly are the last 2 applications, what we can do. So first, because of those Dropletronic devices are modular, we can easily combine them together to form, let's say, we start with a NOT gate. If we combine them together, we'll have an AND gate or an OR gate, depending on if we put them in series or put them in parallel, and therefore we can have beautiful logic circuits by monitoring the 2 inputs and 1 output signal, as shown at the bottom. But more importantly, as I mentioned, the benefit of using Dropletronics is we can try different ions. And in this case, we were inspired by how neurons using neurotransmitters as the signal to form this so-called memory resistive effect. And in this way, we put an additional droplet containing large ions, which is polylysine, which is a charged macromolecule, and it is not bonded to the silk fiber. Basically, the polylysine could be moved by applying an input voltage or current in this case. And therefore, we can use this large molecule to achieve some memory effect. And the process is shown on the right-hand side. So at the beginning, those polylysine is located at the top droplet, as shown here. If we apply a high input voltage or current, it will push those polylysine to insert into the middle, sort of the base droplet here. And because of the reverse charge they have, polylysine is slightly positive charge, and the middle droplet contains negative charge hydrogel. Those two molecules will neutralize each other, and therefore creating this high conductive area. Ions can freely move through this area, and therefore we have high conduction. And also we can apply a reverse voltage or current to withdraw those polylysine. And that needs to be a strong input voltage or current. Just by removing those polylysine will stuck there because they are large molecular size. And therefore, by repeating this circles again and again, we can use this to store memory, as shown in this slide. So as shown here, the black curve is the original IV response. If we insert the polylysine into the middle base droplet, it will make it more conductive or make the conduction signal stronger, as shown here. And if we apply a reverse conditioning current or voltage, it will withdraw those polylysine and make the conduction less, or make the droplet transistor less conductive, as shown here. And the process is fully reversible, and it can be regulated by what polymer we use, right? Polylysine has different molecular weight, and we can use that to actually achieve a short-term memory resistance effect or a long-term memory resistance effect. So as shown here, without using polylysine, let's just use some potassium chloride salt. We didn't observe a strong memory resistive effect, as shown at the bottom of the black curve. And if we slightly increase the molecular weight of polylysine, let's say from 1 to 5 kilodalton to 15 to 30 kilodalton, we did observe a strong increase of the modulation effect. Basically, how large can we change the conduction current as monitored here. However, if we further increase the molecular weight of polylysine from 15 to 30 to 150 to 300 kilodalton, we see the modulation effect, the difference between these two curves, decrease slightly to a middle range, but it's more stable. This is because for larger polylysine, it's very hard for us, or it gets more hard for us to insert them into the middle base droplet by just applying an input voltage or current. But because of their large molecular weight, they will stuck at the middle droplet, as shown on the right-hand side, for a longer time, and therefore we can achieve a real long-term memory storage for up to hours. But then the water evaporation will become a problem. And by changing this molecular weight range, we can tune the memory time we want to achieve from just a short-term potentiation or depression into a long-term plasticity. And this is purely by modulating the ion size of the polylysine, which is also the benefit of using Iontronics with different ions. Lastly, the last application I want to show you is to use biological signals as the basically ion transport from cells and tissues as the modulation signals for our Dropletronic devices. In this case, we collaborated with Professor Christopher in Oxford in the biological department. And basically, they have this human differentiated iPSC cardiomyocytes, and we culture them at the bottom of on top of the petri dish, and then we can use the transistor to actually sense the electrophysiology signals from those iPSC cardiomyocytes. And the way to do that is first we need to encapsulate the Dropletronic transistor into an organogel, which protects the ion leakage from the droplets into the aqueous buffer later on. But we do need to drill a small hole at the bottom, or by connecting the middle base droplet with the cardiomyocytes to let the ions generated or the ionic signal generated from the carbon monoxide to go into the device, and then it will modulate the conduction here, and then we can see the signal by connecting these droplets with external devices. Shown in the middle, this is how the device looks like. We encapsulate, this is very small, I need to mention. The total size is just about hundreds of micrometers. So we need a microscope to actually see this and target it, and then to drill it with some specific tools. But overall, the device is flexible, of course, but it's also very well compatible. We didn't observe any hazardous effects when contacting this Dropletronic device with all types of cells. And this is the benefit of iPSC cardiomyocytes. We can derive that into both atrial or ventricular cardiomyocytes, and they have significantly different beating patterns, which we can monitor them by using Dropletronic transistors, as shown on the right-hand side. So the red curves are the monitor signals by the Dropletronic transistor. The blue curves are the fluorescent volt or the memory potential imaging by fluorescence imaging, which is they have the same signal frequencies. But as you can see here, they do have for our device, it has a delay comparison to fluorescence. One thing is because ions need sometimes to pass through if I can go back, those ions need to pass through those channels, right? From the cell environment, extracellular matrix into the device. So that could take delay the signal transduction. On the other hand side, ions do move slowly by comparison with electrons. So that is an unavoidable challenge for us. But I need to mention this is the first time people can use Dropletronics to directly sense ionic signals from cells and tissues. And that goes to the end of my talk. So discussion about this work, the breakthrough of Dropletronics is we develop an approach to fabricate soft, freestanding, and 3M Iontronics systems. And the 3M stand for microscale. Those devices are very small. Modular, the droplets can be connected very simply by just putting them together. And the multifunctional, because of the hydrogel we use can be modified. We can also incorporate membrane channels and other particles, et cetera, into those droplets. I showed you some examples, the droplet batteries, the lithium-ion batteries, fuel cells, and even we can do the hybrid printing with those droplets. And also later on, how we control the ionic transport. We can use Dropletronic diodes, transistors, memory resistors, or artificial synapse, what do we call it, and to build circuits and biological sensors. There are some caveats at the moment. Water evaporation is a big deal for us. We need to prevent that by either using some more hydrophilic hydrogels or even combined with some organic polymers to achieve this organohydrogel droplets. On the other hand side, another a major people or the second reviewer always asks us what is your performance in comparison with electronics. Of course, the performance need to be improved. But if I could argue a little bit with this, the Iontronic or the Dropletronic area is very new, it's very young, it's just about 10 years old. Electronics is about 50 to 70 years old, with a lot of investment from industry, from academia as well. So we're very young and we need space to improve this. However, I think the future is very bright. We have a very promising future. And this is one of my favorite quote from Professor Bakut from Paris. Basically, the meaning is that if we think about how powerful our brain or any animal's brain is, basically we can use ions, and purely by using ion transport to achieve a very powerful computation or even cognition capabilities, right? Maybe we can mimic that and achieve the same thing with our Iontronic devices. And in my lab, maybe with Dropletronics, we can start with that. So to summarize, Dropletronics is biocompatible, soft, miniature, and Iontronic, obviously. I believe it is a bottom-up technology. And I think this could relate to other synthetic cell experts in the field. Our synthetic cells or droplet-based devices are are bottom up because we can assemble biological system, membrane channels, protein pores, peptide pores into those droplet interface bilayers, and therefore we can use that to replicate some biological capabilities, and we can use that to interface with cells and tissues. In comparison, bioelectronics is more like a top-down technology. People modify electronic devices and make it softer, make it more flexible, and can interface it with cells and tissues. So it's more like a top-down method. So maybe by combining them together or collaborate with each other, we can build this a parallel step to bioelectronics by using Iontronics or Dropletronics. And just at the end, I just highlight to my new lab. We just started in January, and at the moment we are looking for more people to join us. PhD students, for example, with backgrounds in manufacturing, in some material science, or in some bioengineering or organoids are also welcome to talk to me by just sending me an email. With that being said, I come to the end of my talk, and I want to thank you for your listening, and I want to thank all the collaborators I had and the peoples in Oxford, Professor Hagen Bailey, Professor Chris, Professor Ming, and my other collaborators all around the world. So thank you for your attention, and if you have any questions, drop me an email or you can type it into the chat box, which I can see now. So
Yeah, we have a couple in chat already. You already answered mine. I was going to ask about water evaporation, and you touched on that briefly, but there's a couple in the chat. If you could just read them out loud before you answer for anybody watching the video later, that'd be awesome.
yeah, sure. So the first question I can see is from Ben. Thanks so much, Professor Yujia, for your presentation. It's really inspiring from the perspective of bioelectronics. I have a question. In the synthetic cell approach, have you tried to enhance the reaction and conductance inside of the droplets with enzymes? If so, how complex where is it? If so, how complex do you think metabolisms inside of the droplets can be? Thanks. Great question. We do think about how to increase the conduction in the droplets, right? It's because it's hydrogel networks or aqueous solutions. The transport ions is always a challenge or a thing we can improve. For example, we have tried to incorporate liquid metals. It is more electron conductive. But also we try to incorporate, let's say, P-dot PSS polymers is both electron conductive or ion conductive and other proton conductive polymers. We haven't tried actually to think about using enzymes to increase the conduction or the conductance. And yeah, maybe just not my field, or we haven't thought about it. Thank you, Ben. So the second comment from Prasamesh, sorry, I probably pronounced your name wrong, but you said, thank you, Dr. Yuja, for the fantastic talk. How stable are the ion gradients over time? I ask in terms of leakage, whether the lipid bilayers or hydrogel matrices help retain the ions. Also, how is the device performance preserved? I think of real-world biomedical applications. Batch-to-batch variability could be a concern. Yeah. As I mentioned, the stability is a key issue. In our case, for example, if you remember the first application I talked about, those droplet salt gradient batteries, the lipid barrier is very stable if we just keep the droplet network in a lab condition, which is in an incubator with controlled humidity and temperature. It is quite stable. Also, we can further increase the stability to a certain extent by using some synthetic copolymers, which is larger than lipids such as DPHPC, POPC. Some copolymers could improve the stability. However, the water evaporation and eventually, if we translocate the droplet network into an aqueous solution or actually a biological, let's say a physiological condition, those lipids or copolymers will eventually get away into those aqueous solutions, right? So that is also another reason we want to use hydrogels to build droplets, which is more stable in real world applications. But fundamentally, I would say Droplets is a very versatile platform. We can try as my answer in the first question, we can try ionic liquids, polymers, particles, etc. etc. So this problem is there, but I would say we can solve it with further investigation. Okay, so that's I think all the two questions actually in the chat box. Yeah.
Yeah, I'm gonna give a last call. Oh, there might be another one. Yeah.
Okay, another one from Francesco. Thank you for the great talk. I had a quick question. Have you explored actively controlling ion transport in the droplet system? For example, using thermal or pH responsive polymers, membrane pores, or similar approaches? Great question. This is actually very future looking because at the moment in my lab, we have one student actually working on this direction. But I do remember in some previous publications, people used photoresponsive membrane channels to generate this ionic flux in and out from droplets to the aqueous buffer, basically proton fluxes. So I think people have been doing that, and this is also one direction of my current group. Thank you, Francesco.
It's very inspiring work. And like you said, there's so many different directions that it could go. I
Yeah.
am intrigued. I'm excited to see what other like adding enzymes or trying different things too. Yeah, yeah, just trying anything, I guess, is exciting. But hopefully, we'll see you back in however long it takes, and we'll see a progress report. And
Yeah, sure. I would love to. Maybe after 2 or 3 years, I'll come back to the Build a Cell seminar and give a follow-up talk. Hope by
yeah,
that time we can solve many of the issues and the problems. Yeah.
yeah. All right, I'll give our last call for questions and or unmute yourselves if you'd like to. But thank you so much again for giving this wonderful talk. I'm expecting you're going to see a handful of emails regarding those opportunities you posted about. It's wonderful of you. And thank you everyone for attending another Build a Cell seminar.
Yeah. Thank you all. Thanks for the organizers, and thank you everyone for listening and questioning. Thank you all.
Thank you.
Bye bye.